I'm Richard Hodis, the Director of the National Institute on Aging, and welcome you to the Florence Mahoney Lecture on Young Blood for Old Brains. Just to let you know about the Mahoney Lectures, they're a part of the weekly NIH Director Series. These lectures are sponsored by NIA, and they're named in honor of Florence Stephenson Mahoney, who lived from 1899 to 2002. And until the time she died at the age of 103, I would meet with her regularly in her home in Georgetown to discuss the topic. She was a bright, alert woman whose efforts were largely responsible for the founding of the National Institute on Aging. She had great contacts and administrations in Congress, and her place was a salon for discussions that led to a lot of important events at NIH and at NIA and specifically. And when we talked, she was always insistent on wanting to make sure that the lecturer chosen was going to be someone who was going to support rigorous, hard science. She wanted none of this soft nonsense. And so I think she'd be delighted with our lecture for today, honoring the name. And so it honors myself and Florence Mahoney to introduce Tony Wyss-Coray, the T.H. Chen Professor in the Department of Neurology and Neurological Sciences at Stanford, and co-director of the NIH Alzheimer's Disease Research Center there. Dr. Wyss-Coray received his bachelor's, master's, PhD, and postdoctoral fellowship training in Bern, Switzerland. In 1993, he moved to the US to pursue a postdoctoral fellowship at Scripps. He joined Stanford in 2002, following some time at UCSF. His lab is a part of the Glenn Center for Aging at Stanford, Stanford Neurosciences Institute's Brain Rejuvenation Project, and as I said, the NIH Alzheimer's Disease Research Center. He and his team are funded by the American Heart Association AL Initiative, by NIA, and by the NOMIS Foundation. The Wyss-Coray Lab has made enormously important contributions broadly in the field of age-related change, including the degree to which changes in cognitive decline in Alzheimer's disease are relevant to the approach. His team is following up on earlier seminal discoveries, which showed that circulatory factors can modulate function of the brain and structure. That some cases that factors from young blood can positively influence brain function, rejuvenate old brains, and that reciprocally there are some factors from old brain that can be detrimental. It's my pleasure to introduce Tony to you to talk about the latest in his work in this exciting area. Please join me in a warm welcome, even in absence of in-person applause. Tony, look forward to this.
Thank you, Richard. It's a real honor and pleasure to be here and to tell you about our research and what excites us on an everyday basis. And that is really to understand how the young brain, this supercomputer, if you will, becomes susceptible to cognitive dysfunction, neurodegeneration with aging, and how age is in fact the key risk factor for Alzheimer's disease and other age-related neurodegenerative diseases. This is important, we think, because there's really a tsunami of Alzheimer's coming towards us. If you look at the numbers, roughly a third of the people at age 85 or so have Alzheimer's disease. And if you look at how old is the average Alzheimer's patient, they're usually older than 70 or 75 years of age. So it's really a disease of the aged people. And the risk to get the disease as you get older becomes higher and higher. And there's an estimated 75 million people just in 9 years or so that will have this disease worldwide. And that is because people are getting older across the globe, not just in the industrialized nations, but really people are getting older. And that's, of course, a wonderful thing. But as we get older, it's not just Alzheimer's disease and neurodegenerative diseases that increase, but it's really a number of diseases. And in fact, the diseases that are plaguing us the most prevalent diseases are all age-related. And this graph here shows the number of diseases that afflict people on the x-axis from young to old. And you can see at around age 65 or so, the average person has 2 age-related diseases, and that keeps increasing rapidly as you get older, people having multiple diseases. So that started to prompt many people in the aging field, and especially at the NIH, where this Geroscience initiative was formed, to ask the question, what is aging and why does age contribute to all these diseases? And could we possibly slow down or reverse aging to tackle all these diseases? Rather than tackling one at a time, what if you could affect the aging process directly and reverse aging in what often is called rejuvenation? And that's, of course, a dream that humanity has had probably since they could communicate with each other, if there was some way to stay young. And in this famous painting from Lucas Cranach the Elder in the 16th century shows one rendition of this fantasy, if you will, of old people entering a fountain of youth and re-emerging young and rejuvenated on the other side. Now, we know, of course, that there is no such magic fountain. There's no magic potion that could achieve that in humans. But over the past 10, 15 years or so, it has been increasingly become apparent that in mice, there might actually be a possibility for a fountain of youth, and that rejuvenation may be possible, even in complex organisms. And in mice in particular, many different groups have now shown that metabolic interventions such as caloric restriction, exercise, or small molecules that target metabolism and nutrition can slow down or reverse aspects of aging, not just one organ, but multiple organs often. And mice live longer or show clear signs of rejuvenation. Senescent cells can be removed and have the same effect. Transient epigenetic reprogramming, where transcription factors that were initially described by Shinya Yamanaka, rather than reprogramming cells to an induced pluripotent stem cell, you reprogram only a little bit, turn the factors off, turn them on and off multiple times. And what that seems to induce is a rejuvenation of tissues, erasing some of the epigenetic marks of aging. And then lastly, and this is not an exclusive list, this concept, this idea that factors in the circulation in blood may have rejuvenating effects on the body. And what I want to discuss with you is really this paradigm by which young factors can rejuvenate mice, and how that could potentially teach us about the aging process in particular, and how we could eventually harness that to have these desired effects of rejuvenating, regenerating tissues. And so this started really with a paradigm that is called parabiosis, in which two animals are sutured together, a paradigm that is over 100 years old and led to important insight into transplant rejection, but also hormone regulation and metabolism. And this model called parabiosis has been used in more recent time to study aging. And in particular, Tom Rando here at Stanford asked the question whether an aged muscle could be regenerated and rejuvenated with this model. And so when a tissue or when a mouse gets older, the muscle and specifically its stem cells cannot regenerate well, and they can't regenerate an injured muscle. And Tom asked a simple question: is this because there is a defect in the stem cell, intrinsic defect, or is maybe a factor from the environment missing? And so he paired young with old cells, and this was done by two of his fellows at the time, Irina and Mike Conboy. And so they paired these mice, and what they found is that the old muscle could indeed be regenerated and rejuvenated, almost functioning like a young muscle in these pairs of mice. They also noted regenerating effects on the liver, and they had some indication that there was increased proliferation of some cells in the brain. And that's where a very productive collaboration between Tom's lab and my lab took place, and we started to explore this idea that the brain could potentially be regenerated and rejuvenated. But beyond that, increasing number of labs has shown that the pancreas, heart, bone, aorta, and many other tissues, a growing number of tissues, benefit from this exposure to young blood in this model as well. What my lab was able to show, and specifically Solvilate, I'll show you in a minute, is that you actually don't need to suture the mice together. You can simply collect plasma from either young mice or old mice and repeatedly infuse it into the other mouse. And why would this potentially work? This clearly sounds too good to be true. And certainly when I talked about this initially, there were a lot of sort of question marks on people's faces. But if you think about what the blood actually does, the blood is, of course, transporting immune cells. It transports red blood cells that carry oxygen to all the tissues, but it also serves sort of as a highway of communication, if you will, between all the tissues in our body. And so this way, tissues can communicate with each other, and the blood carries all these different factors. And as we'll see, as blood ages, the composition of these factors changes. And it's so maybe less surprising to you after this talk that this might actually work, at least to some extent. So we asked them a specific question: how does the composition of the blood change with age? And this is the culmination of many different experiments that we did over the years in a really fruitful collaboration with Adam Butterworth at the interval cohort and near Barcelona with longevity cohorts, where we had access to plasma samples from over 4,000 healthy people aged 20 to 100. And we were able to measure 3,000 proteins in this experiment with a platform called SomaLogic that uses something not like antibodies, but specific modified oligonucleotides as specific reagents to measure the levels of these thousands of different proteins. And when you look at this experiment, so this is basically the data from this whole study, and we have here the 4,000 people on the x-axis, the 3,000 protein on the y-axis. You can see that the levels of these proteins shown in blue, low levels of a protein, in yellow, high levels of a protein, they change dramatically as we get older. You also notice that the changes are not linear. They come sort of almost randomly. But when we actually ask at each age how many proteins change between younger people and older people, and we count the number of proteins that change at each given age, what we discovered were these almost waves of aging, where you have, for example, here you have about 300 proteins at age 35 or so that are significantly different between people who are younger than 35 compared to those older than 35. Another wave around age 65, and then the largest wave at around 80 years of age. So this again makes the point that age is not synchronous, but it also makes us wonder, what are these waves of aging? What are the proteins that change here? We know that some of the proteins are present in every wave because they keep on changing as we get older, but some are very specific to these changes that occur early on in life. And so we're exploring now whether these changes here on an individual level actually predict how you're going to age down the line, and specifically what type of organs and tissues may age. But much more work to be done here. But coming back now to the parabiosis model, and again, I mentioned Saul Vileda already, who has now his own lab at UCSF. Saul really took the question of how could the brain potentially benefit from exposure to young or old blood into this paradigm that is described in these papers. And what Saul did is, beyond the parabiosis, he collected actually blood from young mice, got rid of the cells so that he's left only with what is called plasma, injected that repeatedly into old mice, and he found that this led to increased stem cell activity, increased plasticity, less inflammation, and most importantly, improved function, memory, and with a number of different paradigms. What Saul also noticed for the first time is that the young mouse actually suffers from this exposure to old in the parabiosis model, but also could again be mimicked by transferring plasma. So here, Saul collected plasma from old mice and repeatedly infused them intravenously into young mice, and that led to the exact opposite effects. It reduced neurogenesis, reduced plasticity, memory function, and caused more inflammation. And a number of other labs have recently been able to show, for example, Gerard Carcenti and Eric Kandel could show that the same, they could repeat that young plasma has these beneficial effects, and they could actually show a specific factor that is able to do that, in this case, osteocalcin. Other factors have been put forth to have beneficial effects either on the brain or on other tissues, some of them listed here. And then what we also discovered is these age-promoting factors that apparently have detrimental effects and are in part responsible for the negative effects. So these are examples of proteins in the blood that change with age, that have either beneficial or detrimental function. We think there are many factors, it's not a single one, and this is certainly not an exclusive or a final list, by no means. But because this is so translatable, and plasma has of course been given to people for decades in injury settings, after accidents, but also for therapeutic purposes, and often even fractions of plasma, not the entire plasma, but just a component of the plasma has been produced and then administered. And so we started a company that I disclosed earlier on and could indeed show that this treatment is safe in Alzheimer's disease. You can either infuse whole plasma donations from individuals, or more recently, components, a fraction only of plasma that still contains a few hundred proteins. You can infuse that repeatedly. It is safe, and we had some evidence that this might even be beneficial, although these are not yet placebo-controlled trials. But interestingly, an independent group pursued the idea that there are maybe detrimental factors accumulating in Alzheimer's disease in the blood with age. And so what they did is they removed plasma from patients in a process called plasmapheresis. And so patients go into these almost like a dialysis center where their blood gets exchanged with a fraction that contains albumin, as well as a few hundred other proteins. And what they showed actually in a phase 2b and phase 3 trial in Europe, that was completely blinded, placebo-controlled in 500 patients with Alzheimer's disease, that this is in fact beneficial, that these patients are doing better, especially those with moderate disease. They have reduced progression of the disease, and they're improving in memory and daily functioning. So here, again, what this group did is they remove aged plasma, and they give a fraction of albumin that incidentally comes actually from young people, because the average donor is 35 years of age. So this is slowly, I think, getting to a validation of this approach that we see in mice to translate that into humans. For the future, and we can discuss this after the talk maybe a bit more, I think we do need to understand what the components are here to really make this a treatment for everybody. But to me, this is a sign of a experimental medicine, if you will, that this might be an approach that is worth pursuing. But again, coming back to this question, this is almost too good to be true. So how would this potentially work? And a lot of questions have come up here out of these observations that I think we need to tackle, we want to tackle because they're so exciting and interesting. One of them is, of course, what are the factors? Where do they come from? Which cells in the body produce the good factors or the bad factors? And then very related to this, what are the targets of these factors? What are the cell types and tissues that benefit, for example, from young blood? How do the factors reach the brain, especially for a question like Alzheimer's disease? We have this blood-brain barrier that we have been taught doesn't let things into the brain. So how would this potentially work? And then a question that is more conceptual and related to the study of aging is, do actually all cells and tissue age at the same pace in our body? Or do they have different trajectories, different clocks, if you will? And what I showed you with the plasma already suggests that they may not all age at the same time. And then related to that, and this is also related to the concept of senescent cells that Judith Campisi has put forth, that some of these cells may actually age faster and they secrete detrimental factors that could potentially even spread aging through the body. So I want to give you two vignettes of questions that we're addressing from this group of questions here. And the first one is, which cell types does young blood actually rejuvenate? And for this, we first wanted to tackle, how do tissues and cells actually age in the organism? And this was spearheaded by Nick Schaum in my lab, who asked this somewhat naive question for his PhD thesis: where do these factors that we see in the blood change? Where are they being produced? And we decided to simply do transcriptional profiling to see where are the genes expressed. And initially, we planned to do this at the bulk level. And we talked to Steve Quake here at Stanford and asked him if he would be interested to collaborate. We started to explore this idea of young versus old transcriptional level. Luckily, by that time, Steve became the co-director of the Chen Zuckerberg Initiative and had the possibility to do this at the single cell level. And so we embarked by recruiting over a dozen different groups here at Stanford and UCSF to really harvest all these tissues, that dissociate them into single cell, and then do single cell RNA sequencing. And that led first to a map that just described gene expression across all major cell types in the mouse, and then how they age how they age at the single cell level. And then this last paper here, a combination of using bulk RNA sequencing with single cell, which I think is the most powerful approach, because the bulk gives you the confidence, the power to look at all genes, and then the single cell signatures allow you to extract where does that signature come from. And so just as an example, on a bulk level, looking at all RNAs in one of the fastest aging tissues, gonadal adipose tissue, you can see here how these genes change from young mice to old mice. And you can again see that the changes are nonlinear across a lifespan. When you look at all the different tissues in the mouse that we had the opportunity to study, you see that with aging, an increasing number of genes are downregulated or upregulated, but by no means is this a homogeneous process or a synchronized process. It seems some tissues age very late, some show changes earlier on. And that really led to a concept that was first put forth by Monica Driscoll in C. elegans almost 20 years ago, when she noticed at the EM level that tissues in an individual worm, in individual worms, show different signs of aging. But with this transcriptional approach now from our group and other groups, it really has become quite clear that tissues seem to age on different trajectories, that you have some that show profound transcriptional changes during the reproductive phase, different times of the reproductive phase, and some only start to show significant changes later on at the post-reproductive phase. But what you also see, and this is not shown here, that from individual to individual, there is variability when the onset of aging starts in these tissues. And then you can, of course, look at this at the cellular level. And so I want to discuss with you this graph here, which we call a Picasso plot, which shows in these different tissues that are shown here on the x-axis and individual cell types in these tissues. And so we look at the number of genes that are significantly changed in each cell type from young to old. And the size of the dot reflects the number of genes that change in each cell type with age. So a large dot means a lot of genes change with age, small dot, very few genes change. The lines that connect these dots indicate how similar the top genes are. In a way, are these cell types aging at a similar way? And what we noticed are two primary observations, and certainly this first one is, I think, a very solid one. So here, for example, for stromal cells, no matter in which tissue they are, they seem to share the transcriptional profile of aging. So no matter in which tissue the stromal cells are, the top genes that change with age are the same. The same for endothelial cells. So endothelial cells, even brain endothelial cells, though they are very different from other endothelial cells, they share common signature in the top age-related genes across all the major tissues. And then we saw something else also. We saw these tissues where apparently cells of different types, different cell types, certainly in the skin, have a shared signature, suggesting possibly, and we don't have really that strong evidence yet, but maybe here the tissue itself is actually driving the aging process, rather than what you see here, maybe a cell-intrinsic aging process. But more work needs to be done to really firm this up. So this was very exciting to us. And this is just looking at normal aging. So what I want to discuss with you now is the aging at the cellular level in this parabiosis model. And so again, to give you sort of an orientation, we have young versus age, normal aging, and we call this age. Then we have in parabiosis models, we always have pairs of young mice. They're called isochronic young. We have pairs of old mice, isochronic old. And then we can look what we call accelerated aging. So how does the young mouse exposed to old, how does that gene profile look? And we can look at rejuvenation by looking at the old mouse and how its gene profile changes in response to a young circulation. So we have three groups. Age, which is sort of the benchmark, the ground truth, if you will. Accelerated aging, so does exposure to old mimic aging? And then rejuvenation, does that reverse aging? And so again, we look first at just aging. And here I show you 18 out of 47 cell types that we had the opportunity to analyze here. So here you just see the number of significantly different genes in these different cell types, and they're ranked by number of genes. So you see hepatocytes are the winners. They show the most age-related changes. But let's look now at accelerated aging first. Here we have a different order. So it seems that some of these cells, you have more than 1,000 genes that change and show age-related changes that are induced by an old environment. So these genes give you a ranking of which cells are most susceptible in this young mouse to an aged environment. And then vice versa, you can ask what cell types are changing their signature in the old mice when they get exposed to young blood. And here again, we see some cells show 1,000 genes that change simply in response to being exposed to this young blood in the parabiosis pair. And what you also note is the ranking is different. You have hematopoietic stem cells here at the top. They're a bit further down in accelerated aging. And then you have a number of endothelial cells, for example, that are ranking relatively high in the accelerated aging paradigm, but they don't show up here in rejuvenation, suggesting, which we have actually described in an independent separate study, that endothelial cells are highly susceptible to an aged environment. But now you ask probably, well, are these the same gene here that change with normal aging? Not all of them. We see, again, big differences from cell type to cell type. These SMCs, the smooth muscle cells, these show almost 50% of the genes that change with age are actually identical to those that change with normal aging. Stromal cells are also showing a lot of changes that are consistent with normal aging, as do fibroblasts. When we look at the opposite of which genes can be reversed, it's a much smaller percentage, but still potentially significant and important. And of course, we want to know which ones. And when you look at this, you can see, for example, the hepatocytes, which show these dramatic changes with age. Many of these genes can actually be reversed with exposure to young blood. So they go in the opposite direction, significantly in the opposite direction. And just give you a flavor of two cell types, here we look at gonadal fat stromal cells in the accelerated aging paradigm, and to look at not just number of genes, but here we look at a volcano plot. So here, genes that go up with aging, genes that go up with accelerated aging or go down. And then here is the p-value, so a classic volcano plot. And now we can compare how do they relate to normal aging. So here we have normal aging, reverse, go up with normal aging, go down with normal aging. And then here we have the accelerated aging paradigm. And what you can see is that a majority of the genes here in this cell type are actually going consistent with normal aging. And the genes in this network, many of them are in matrix and collagen metabolism or collagen structure and function, potentially consistent with some of the observations that have been made in the aging field. If we look at the hepatocytes, which I mentioned to you, show this rejuvenation signature in the liver, we see again in the volcano plot genes that go up or down with this paradigm. And here you see the majority of genes in this particular cell type go up with rejuvenation. And if we look again, compare this to normal aging, we see that indeed many of these genes are reversed with aging. So if you rejuvenate here with young blood, the genes go in the opposite direction than they would go with aging. And really interesting, many of these genes are in mitochondrial metabolism, electron chain transport, a theme that we see general if we look at the organismal level of this parabiosis effect. So much more work needs to be done here, but you can see that this could be really a rich resource in trying to understand what are molecular signatures at the single cell level, at the single cell type level that benefit from exposure to young blood or show a similar trajectory that you see with normal aging. And I think these signatures can be informal for us to then find mimetics, whether these are drugs or other interventions that show the same behavior that would rejuvenate the cell or accelerate the aging of a cell. And in that context, there was a recent paper that actually used a caloric restriction to do a similar single cell profiling. And they looked specifically at rats that were either fed ad libitum, and when they get old, they gain a lot of weight. And if they're on caloric restriction, they don't, and they show all these health benefits. And so at the single cell level, they looked at 6 tissue that were pooled from a number of rats. And what they find is, again, that individual cell types show very distinct responses to this intervention, to this beneficial intervention. And they can start to tease out specific signatures of rejuvenation. And what one of my fellows is now doing is to compare actually some of these signatures that caloric restriction has at the single cell level with the ones that young blood has to potentially identify common signatures of rejuvenation and identify what are the cell types that most benefit from this intervention. So to summarize this part, what we find is that tissue transcriptomes reveal different rates of aging. Cellular aging signatures are driven by tissue or cell type, at least that's what the sort of a first conclusion we have. And then cell types respond in quite distinct, specific ways to changes in blood plasma. And parabiosis elicits common transcriptional signatures of aging and rejuvenation, implicating sort of at a first global glance, the extracellular matrix, as well as oxidative phosphorylation, respectively. So for the remainder of the talk, I want to discuss with you this question of how could this potentially help the brain with this notion of the blood-brain barrier. And this is a project that Andrew Yang, a graduate student, tackled. He's since graduated and is wrapping up some studies in the lab. But basically what Andrew asked is this question that I just posed: how would these proteins that we see change with age, how would they potentially induce the benefits or the detrimental effects in neurons, microglia, astrocytes, or stem cells in the brain? And you can imagine sort of three simple concepts. One is that you have areas where this blood-brain barrier is actually not tight, where there are no tight junctions, where factors could have direct access to the brain tissue. Then there's a possibility that is well documented with the transferrin shuttle, how it's often called, where a protein, in this case transferrin, binds to a receptor that then gets internalized, carries the cargo on the abluminal side and releases transferrin. So this receptor-mediated active transport is certainly a possibility. And then lastly, we would have the possibility that there is an activation of endothelial cells that changes their behavior such that they communicate now in different ways with cells of the vasculature, but also the end feed of astrocytes. And that would change potentially neurovascular coupling and other downstream effects, and eventually regulate neuronal function. Now, traditionally, the integrity, sort of the functionality, at least from a simple mechanistic, mechanical view, was studied with tracers that include horseradish peroxidase, an enzyme from plant, Evans blue, which binds to albumin, or polymers of dextran of various sizes. So people inject these into the blood, and in a young, healthy animal, these tracers would not get into the brain, and that actually led to this concept of the blood-brain barrier. But if there is a rupture because of an injury or with aging, age-related changes of various causes, some of these tracers would then leak into the blood, leading to this concept of leakiness of the barrier or disruptive blood-brain barrier. Now, that's, of course, I'm not trying to dismiss any of these key studies that have been done to come up with the whole concept of the barrier and how it's structured and how it functions. But for our specific question, it of course doesn't help us to understand how these thousands of proteins that we can measure, how they could potentially communicate with the brain. And there are indeed proteins of many different sizes in plasma. We can measure now over 10,000 proteins. Some of them are water soluble, some are lipid bound. Many of them are in vesicles or even lipoprotein particles. So a very complex mixture of proteins. How do they interact with this barrier? And how would they potentially exert these positive or negative effects that we can observe? So Andrew took a very simple approach, a somewhat trivial approach, if you will. He isolated the plasma proteins from young mice for all the studies that I'm going to show you, and labeled them with a number of different tags, if you will. Classic radio labeling, fluorophores of different charge and sizes, different intensity, different colors, biotin, but then also bioorthogonal labeling, for which we actually got support from a Pioneer Award from the NIH to label the whole proteome of a mouse so that it has little tags on the proteins that we can then detect. Once we inject them in a mouse, we can detect any of these tracers, presumably, and we can figure out whether they get into the brain or not. And for that, we used then classic radiotracer methods. We used histology. We isolated cells and did next generation sequencing, as you'll see in a minute. And for all these experiments that I'm going to show you, the basic paradigm is we label the proteins, we inject them IV, and then 4 hours later, so very acutely, we extensively perfuse the brain. So we wash the vasculature for many minutes, and then we take out the brain and we process this whichever way we want to do this. Sometimes we remove antibodies and albumin, the most dominant proteins in plasma, so that we have a better chance of detecting some of the other proteins, which is shown here. So albumin and antibodies make up the majority of the proteins. So if we want to focus more on the other ones, it helps to deplete these. And so this is just an example of a brain that was taken out from a mouse that was just treated like I told you. And then we cleared it with the iDisco method that Marc Dessier-Levin described. And then we imaged this brain. And what you can see is that there's a lot of label left. So here we use the fluorescent tag to label basically every protein on average in the plasma. And you see there's a lot of plasma in here. So if we use more classic histology, a coronal section from a brain, we can see that there's an extensive, exquisite labeling of the whole vascular tree. We also see these areas, so plasma is white here. We also see these large areas, which are the ventricles. So these are the spaces where the cerebrospinal fluid is. And here we see the hypothalamic regions where we know the blood-brain barrier is actually not tight. There are openings that allow some of the proteins to get in. So consistent with this tracer idea. But if you use one of the tracers, the classic tracers, you would see nothing at all here. So horseradish peroxidase would not label the blood vessels. Dextran would not label the blood vessels. So what is going on here? If we zoom in, we actually see that the whole vascular tree is full with these little dots, these little vesicles that you see all across the blood vessels. And what we also see in some areas, we see neurons that are filled with plasma proteins here in the hippocampus or the neocortex. And we see also other cell types. They're quite rare, but here's an example of a microglia that has plasma proteins taken up. And one of my fellows is now isolating these microglia. It's a small portion. There's a different distribution, different brain regions, has different numbers of these positive microglia, but they average anywhere from 1% to 10% across the brain that take up plasma proteins. And now we want to know what proteins are being taken up and what is special about these microglia. What might they be doing with these proteins? But to get at this first with respect to the vasculature, we took advantage of the single cell sequencing paradigm again, and specifically also of the fact that the labeling is actually not even. So if you look here at an example of a blood vessels, these little dots, these vesicles are not completely evenly distributed. So what we did is we injected labeled plasma, we isolated the vasculature first, then dissociated it into individual endothelial cells. And now we have, for each endothelial cell, we can measure with flow cytometry how much fluorescent plasma protein is in each cell. And then we can sequence the RNA transcripts for each cell. So we can correlate gene expression with uptake of plasma proteins, which is called index sorting. Very powerful technique. And when we look at this and correlate gene expression with plasma protein uptake, what we see is this beautiful bell-shaped distribution curve where most genes do not affect uptake of proteins into the endothelial cells, and they're here, it's shaded in gray. But you see here this trace of protein or genes, and there are actually several hundred that correlate, some of them very highly, with uptake of plasma proteins, and some of them are very well-known players in protein transport, including ApoE, which is of course a lipoprotein, but then and LRP8, which is involved in lipoprotein transport, but also most prominently the protein carrier machine that I mentioned to you before, the transferrin receptor, which carries transferrin at very high efficiency and rate into the brain. And then other well-known so-called solute carriers. Some of them are actually being exploited now, the transferrin shuttle or this SLC3A2 to carry specific drugs into the brain using these. And you can imagine that there may be many other such transporters hidden here. What we also noticed, and I think this is something novel, is that on the left-hand side here, we have genes that are negatively correlated with uptake. And we're particularly interested in to see how they regulate protein transport. When we look in what categories are these genes, they make a lot of sense. So the positive correlates are in solute carrier, membrane transport, metabolism, vesicle transport. And interestingly, these anti-correlates are involved in calcification, ossification, matrix degradation, and we have actually evidence that some of these may be involved in calcification of the vasculature with age. And that brings me to the very last couple of slides. How does this protein transport actually change with age? And so if we use the classic tracer method, we label here an immunoglobulin with a radio tag. As many others have reported, there is little uptake into a young brain. You see here mostly labeled in the ventricles, and with age, there is a significant increase in uptake. So we expected that there's also more uptake of plasma proteins into the brain if it follows the same logic of sort of the barrier gets leaky and you get more into the brain. But we saw the exact opposite. We saw an almost 50% reduction in uptake of plasma proteins. And to look at that a bit more carefully, we want to focus on the two major pathways of vesicle-mediated transport. So I mentioned to you already a specific one that uses clathrin to take up receptors with their ligand and shuttle them. And the other one uses caveolin, and that's less specific. It's often called adsorptive transcytosis. And here you have proteins that bind to the cell surface, are then taken up into these vesicles and transported. This is actually the main mode of transportation in the endothelium across the body. But in the brain, this is reduced by about 90% with a gene called MFSD2A that Zhenghua Gu at Harvard discovered a few years ago. So this here is only 10% in a normal healthy brain. And this is most of the plasma transport, most likely what we conclude from our studies. So with age, what we see is that there is a decrease in gene expression for the specific transporter, and there's an increase for genes encoding for the non-specific. And when we look at histological, with histology, and look specifically at clathrin, we see lots of vesicles in young blood vessels, and we see much fewer, as you can see here, in aged. And we see the exact opposite with the non-specific caveolin vesicle, where you have very few in young blood vessels and many more with aging, a several-fold increase, suggesting a shift from specific to non-specific transport. And we can actually confirm that when we look at specific cargo, such as transferrin or leptin, they decrease. So they're specific, they decrease with age. And albumin or horseradish peroxidase, they actually start to leak in, as people call it. So non-specific increases slightly, but the specific decreases dramatically. And because we see less plasma protein uptake, we would argue that most plasma protein uptake we measure involves specific transport mechanisms. And so with that, I like to conclude that we have a view of the vasculature, of the transport of the vasculature, that suggests that there's a shift from specific to non-specific transport. We also note, like Betsa-Slokovic and others have stated, that there's a profound loss of pericytes. And then we actually also discovered this calcification rate, which may involve some of these negative regulators of protein uptake, that with age they become overexpressed and they probably regulate down transport of proteins. And with that, we support sort of this increasing view that the barrier is not really a barrier in the classic sense that is open or closed, but it's more a licensed access that shifts and changes in very sophisticated ways with aging and likely also with disease. And so we conclude that many plasma proteins can enter the young brain using specific transport and are taken up by neurons and other brain cells. And with age, that transport shifts from specific to non-specific, and there is less protein uptake overall. Of course, we want to understand the physiological meaning of that, which proteins go in, what are they doing, and are they somehow involved in disease. And with that, I'd like to thank a terrific team. I mentioned some of them who contributed, also collaborators, mentioned Steve Quaid, but many others who supported us and worked closely with us. And then funding, of course, from the NIH, both for regular funding, but then also the Pioneer Award, which really let us explore these sort of crazy ideas and from the other sources listed here. Thank you very much.
Tony, thank you for a spectacular presentation. I know by count there's a large audience out there, and I'm sorry again they can't be here to applaud in person. Let me ask, if I could, a couple of broad questions. Are there any occasions in which parabiosis, transfer of serum or proteins, has had effects the opposite to those that you've emphasized? So from young to old, for example, having adverse effects. You know, one might have thought some of the changes which occur with aging, maybe it's an optimistic view, might be adaptive and not necessarily all negative in consequence.
Yes, that's a fantastic question. I think people haven't reported negative effects, which is sort of a bias in the field in general, I think. But what you can clearly see from the single cell signatures that, first of all, not all cells respond to the exposure to young or old. Only some of, I showed you the top 18. If you could look at all of them, we have maybe 30% that don't show significant transcriptional changes. And what I also showed you, if you look at how is the accelerated aging similar to normal aging, it's only a fraction of the genes that are the same. So I'm pretty convinced that, first of all, the parabiosis model itself will induce transcriptional changes and will have its effect. But even if you could eliminate that, and we're doing an experiment where we're just infusing plasma, there are probably detrimental effects for some factors. And so we need to identify, that's why we want to know what are the beneficial signatures that we could potentially then overlap with caloric restriction, transient reprogramming, and all these other methods, and find, these are the key signatures that we want to mimic with a drug.
One question that was raised in the chat had to do with the relationship of your experimental systems to what happens with, I guess, clinical dialysis. So let me transmit that question, but also ask as a parallel, whether in addition to the proteins that you've been looking at, there are other molecules like metabolites that might be relevant and perhaps even more difficult to analyze.
Yeah. Again, two very good questions. So the plasmapheresis paradigm that is used in humans is not really what we do if we inject aged plasma, but it's somewhat related or even quite similar actually to what Irina Conboy and Mike Conboy published, where they developed this very sophisticated instrument that changes the plasma in a mouse, and then they supply it with saline and albumin. They call it albumin, but it's an albumin fraction. So it's actually the same that humans get. It's called fraction 5 albumin, which contains mostly albumin, but also a few hundred other proteins, growth factors mostly. So I think they report beneficial effects with that as well in the mice. So I think it mimics to some extent what we can do in the mice. And again, the plasma that we give, or the albumin fraction that people give in these paradigms, always comes from young organisms, from young people or from young mice or from young rats. Your question of, proteins or something else. Yeah, I apologize, I forgot to mention this, but we really focused on proteins for convenience because, they're important, of course, but we can manipulate them. We can synthesize them, we can sequence them, we can measure them, and we can genetically manipulate them. But by no means does that mean lipid mediators, other metabolites, small RNAs, or, there's so much in plasma as people get better tools and more reliable tools, and everything changes with age. So I bet you there's lots of stuff in there that could be targeted, and proteins provide one opportunity, I think.
One other question, Tony. What does heterochronic parabiosis do to protein uptake in aged or young brains?
We haven't looked at that yet. So in our model, we started to measure, to study this protein uptake by simply restricting ourselves to plasma from young donors and with a very acute paradigm. But you could, of course, now, as the question suggests, you could take aged plasma, put it into young mice, or young plasma in aged mice and see how that is taken up. And it's most likely, changing as well, but we haven't really looked at that yet.
And if I could ask you one additional question, given your interest in contributions in the area of Alzheimer's disease in particular, have you tried to overlay the changes you've seen on some of what's been revealed at the omics level about changes which occur characteristic in Alzheimer's versus normal brains?
Yeah, so that's something we are very intensely looking now at the single cell level, focusing on the vasculature actually, and there are clearly overlaps and differences, and it's not yet clear, what are the most important ones. It's, some of the papers that have been published to look at single cell signatures from Wyss-Coray, for example, suggest, these new changes in oligodendrocytes that have not really been looked at before. And I think we will see much more of that. We see very profound differences in the vasculature, in vascular cells that we're very excited about. But I think there's so much that needs to be done. And with these new tools, it's really providing unprecedented opportunities.
Tony, let me thank you again for a spectacular presentation, reminding people who were fortunate enough to be in the select group to be able to talk with you at the T, some of our trainees, that they'll be dialing back in. And for the CME code, I've been asked to remind once again, 31302. And I guess, Tony, when everybody else is off, we get a little chance to wrap that.
Yeah.
Thank you so much.
Thank you so much. Thanks for having me.